Okay, so we're going to talk about the histology of skin and just certain functions and stuff that goes in the different layers. So we know the skin is it's the largest organ in the body, and it contributes to about 15 to 20% of our total body weight. Okay, so the skin, we know it's on the outside region, like it covers our whole body. It's got a lot of functions. It could stretch, it's waterproof, things could go in and out of it, we could sweat through it, our hair comes from it. But I'm going to talk about all the different steps right now. So we have three main layers of it. We have the epidermis, the dermis, and the hypodermis. The most important one that we're going to talk about is the epidermis. That's what I have in the middle. He's just going to be superficial, so he's like on the outer region, right? He can kind of be in the nostrils a little bit, which is like a little like inferior, or like inside the body and the ear, but he's mainly on the outer surfaces, right? And he's just composed of stratified squamous and keratinized epithelium. The dermis layer is a deeper layer of the skin, so it goes epidermis, then underneath it is going to be the dermis. The dermis is going to be a little deep, and it's connective tissue. And then the last one's the hypodermis. It depends on the person, honestly. Some people have more than others. It's basically where the fat tissue is stored. That's where adipose tissue is. Okay, so this is how we're going to do it. We're going to start off with the epidermis. The epidermis gets branched into two different regions. He gets branched into keratinocytes and non-keratinocytes, right? So the major part of it is the keratinocytes. This is composed of 85% of our epidermis. Okay, and we have 5 layers. Really, it's 4, but on some regions of the body we have 5. The regions in the body where we have 5 are thick regions. So the palms of my hands, the palms of my feet, right? We need a thick region because we want more skin there, because you don't want like things to pierce yourself or like to go in. The skin is used a lot, right? Okay, so what are the 5 regions? This is going to be, I'm going to use this acronym, and it's going to say, come, let's get sunburned, right? Okay, come, let's get sunburned is going to just stand for 5 different things. C is the first one, superficial. Stratum corneum. Okay, then L is going to stand for stratum lucidium, right? Remember, this is only for thick, though. This is only thick skin. The other 4 are going to be thin skin. Okay, so let me just circle that. So the L right here, this is going to be thick skin. And let's get sunburned. The other letters are going to be for only thin skin. Okay, so then G, thin skin again, stratum granulosum. Okay. S is going to be stratum spinosum. So all of these start with an S, all of these start with stratum, but the second word is the letter that I'm going to use in the abbreviations. So now B for burned, stratum basale. Okay, what do we have to remember? Basale is bottom. The basal layer of skin is the bottom. Okay, so that makes sense for this to be the bottom. So another thing I want to mention about the thick skin is not just this layer. The thick skin is composed of every single layer. It just has an additional layer in it. Okay, so I'm going to talk about the skin, the keratinocytes, from the bottom going up, because I think it makes more sense to explain it that way. So the first one, stratum basale, right? This is going to be a single layer of small, mitotically active basal cells, and they attach to the basal lamina using hemidesmosomes and to neighboring cells using desmosomes. Okay, what does this mean? Here, this black region right here, this whole box that I have drawn, right? This is going to be the epidermis. So this area is the epidermis, and this bottom region right here is going to be the dermis. So just remember that. Okay, so the basal lamina is the bottom region. It's going to be the last layer, right? The last layer before you enter into the dermis is the basal lamina. So the stratum basale, I said, it's a single layer. So we have a single layer of mitotically active, so able to divide very fast. So mitotically active cells, right? They're called basal cells, and they're attached to the basal lamina via hemidesmosomes right here. This is the hemidesmosome it's like an anchor. It's anchoring down into the basal lamina, and it's anchoring into the basal cell. So it's connecting them together. And then it attaches to other cells using desmosomes. Desmosomes is just like a hemidesmosome, but instead of attaching from like the cell to the basal layer, it attaches from cell to cell. That's what a desmosome does. Okay, now, the next layer I want to talk about is going to be the stratum spinosum. So he's a prickle-like cell which is meaning he's got like a lot of like finger projections coming off of him. And he has several layers of larger keratinocytes that are attached to each other by desmosomes, located at the end of the cytoplasmic processes. Okay, so what does this mean? Prickled cells, finger-like projections, right? I have these little like prickly things coming off of them, right? So this increases the surface area. Why do we want to increase surface area? So you want your skin to be strong, right? You don't want your skin to be loose and weak. And in order to make it strong, you want more connections. In order to increase the connections of the layers of skin, we have these finger-like projections that are going to form desmosomes. More desmosomes are going to be attached to every single cell. The more desmosomes we have, the more attachments we have, right? If I have my fingers like this, one desmosome on each side, it's kind of easy to pull apart. But if I have 4 desmosomes attached, I can't really pull my hands apart, like they're interlocked. So the more desmosomes you have, the stronger it's going to be. And that's what we want. We want our skin to be strong. Okay, so the desmosomes add mechanical strength, like I said. Now, one thing I want to mention before I move on to the other layers. What happens if we have a loss of hemidesmosomes? Okay, so a loss of hemidesmosomes would cause what? That would mean a loss of these, right? This little connection layer. If you have a loss of the connection layer, just think of the basale, the skin, getting ripped off, right? It's pulling off of the basal lamina. If that happens, you're gonna have like a little like space, right? This little gap. In this little gap, we're gonna have fluid. Like fluid's gonna flow in and build up. What's that fluid? That's what's going to be like a blister. That's what blisters are. Fluid getting underneath the basal layer, but above the basal lamina. That fluid buildup is called a blister. Now, if we have a loss of a desmosomes are connecting cells to cells, right? Basal, spinosum, right? If we have a loss of these, what are we going to have? These cells are going to start to separate from each other, right? And we're just going to form random vesicles. Random vesicles could have really bad functions, different types of diseases, etc. So a detachment or a degradation loss of desmosomes is going to cause the vesicle formation, and a loss of hemidesmosomes is going to cause blisters. Okay, now we have B done, we have the basale done, we have spinosum done, right? Let's go with J now. Stratum granulosum. Okay, he's basophilic, so he's like a little darker than the other ones, right? And he's basically all he is a flattened keratinocyte. So he's a flattened one of these cells. The basale and the spinosum kind of like boxes, right? Remember that? Or circles, right? The granulosum is going to be flattened. Okay, what does this mean? He's going to have 2 different branches. He's going to branch into either keratohyalin granules, right? And he's going to branch into lamellar bodies. Okay, the keratohyalin granules, right? This is going to be a precursor to filaggrin. Filaggrin is something that aggregates cells together. So they help the cells join to each other, right? And then a lamellar body is going to be a lipid, a phospholipid, or a cerebramide. This is going to secrete lipids, and these lipids are responsible for an epidermal water barrier. So let's explain it. The first one we have keratohyalin granules. They're just going to help the cells, right, clump together more. So if we're clumping together more, like this, we're going to need protection, right? We're not going to allow stuff like air or molecules or like bacteria to enter in or out of the cell. We need a prevention layer or else we could get sick very easily. The next layer, or next type of cell in this layer, I said, was the lamellar bodies. So when they release like lipids and stuff, we have to remember what the function of a lipid is. Lipids, right? Say we have a little phospholipid bilayer, right? Like that. We have phospholipid tails, whatever. This forms a permeable barrier, right? Water molecules can't enter. Water molecules can't enter this barrier. So these lipids do the same thing. You know, if you put water and oil in like a pot or something together, the water and oil doesn't bind. Same exact scenario. The water is not able to enter through the cell because the lipid is being secreted. Okay. Now, what happens if we were going to have a loss of filaggrin? So filaggrin was for the keratohyalin granules, the first one I talked about, how the cells aggregate together. If you have a loss of this protein or the fibrinogen, he's going to expose the body to the outside. The body is exposed now. Things can enter in or things can enter out. This is very dangerous. Another thing is if we have a loss of lamellar bodies, random water could just flow into our body, but more importantly, water could just get leaked out of our body. We could just leak out water, we'll become super dehydrated and we'll just become nothing. Because we know water makes up 70% of our bodies, this is extremely dangerous if that was the case. Okay, so we did B, we did S, now we did G. So stratum lucidium, all he is just layers upon layers upon layers of dead cells, right? All he is layers of cells that are making my palms thicker. Why would you want your palms thicker? Because this region is things that are touching stuff all the time. If you're constantly touching stuff, you're always losing cells. You're like shedding. The back of my hands, my legs, you're not really losing cells all the time. You're not really touching things. But your feet is constantly, there's always friction on your feet and there's always friction on your hands. So you want more layers. That's why this is thick skin. Okay, lucidium's done. Now we're going to do the last one, stratum corneum. This is the most superficial one. He's squamous layer, but what's important about him? He loses his nuclei, right? He has no nuclei in him, and he's entirely filled up with keratin filaments. So he's only keratin, and he's thin, right? These keratin filaments are just thin flakes of this epithelial tissue. So I'm going to have him all the way at the top here, right? And we have hundreds upon hundreds of these tissues, just no nuclei, it's dead skin. A continuation of corneum, so we could easy to remember, is your hair follicles, it's just dead skin, and your nails. The tips of your nails is just dead skin. It's just growing off. This is dead skin. Okay, so now, these are the keratinocytes, right? They basically just provide structure and not too much function, but some function of the barriers for the epidermis. Now, the other 15% is the non-keratinocytes, right? This is 15% of the skin makeup. We have three different types of important cells here. The first one we have is a melanocyte. Melanocytes are very important because they're pigment-producing cells of the epidermis. So this is where he's located, right? He's going to be in the basal area, right? So the stratum basale. And I have him brown because, how you tan. He comes up, right, into the spinosum. So his cell originates in the in the stratum basale, but his branches lead up into the spinosum. What does he do? Okay, he has a special enzyme on him called tyrosine enzyme. Tyrosine enzyme creates melanosomes. Melanosomes is then going to release melanin content. Melanin content is going to do what? It's going to go into neighboring keratinocytes. So what's a neighboring keratinocyte? These all are the keratinocytes. Here's all the keratinocytes, right? So you see how I have this branch coming up? The melanin is getting released off of these branches via melanosomes, and he's just putting melanin into all the cells, right? Why is this important? It's not just important to get tan. The main reason is because it's going to accumulate this little cap, right? It's going to accumulate a cap called a supranuclear cap. Okay, what does this mean? So the neighboring keratinocytes get the melanin. The melanin is inserted near the nucleus. Remember that. If it's inserted near the nucleus, an accumulation leads to a supranuclear cap. This supranuclear cap is going to cover the nucleus. It's going to shade it. It's going to act like an umbrella, right? And why would that be important? Because if we have a lot of UV light, right, and sun attacking the nucleus, what do we form? We form thymine-thymine dimers. What are thymine-thymine dimers? This is basically the origin of skin cancers. We have T's connecting to T's. This is not good in DNA replication. And where is DNA replication or transcription and translation? It's in the nucleus. So this supranuclear cap is very important because he's going to shade over the nucleus. Okay, now, fun fact about melanocytes, every single race has the same amount of melanocytes. Just the rate they're expressed is differently. So if you're living in a region that has more sun, you're going to increase your rate of melanocytes, right? But you're increasing your rate of melanocytes, so you increase your rate of what? Supranuclear caps. If you have more supranuclear caps or darker, you could shade the sun a lot better, which will protect your DNA. Also, birthmarks. Birthmarks come from melanocytes. Why? So if a melanocyte, right, this brown region is placed in the basale where it should be on the bottom, you're not going to have like little, your skin's going to be normal. But if you have like a little birthmark, like I have a little dot here, right, the melanocyte is actually placed higher up in the skin. If it's placed higher up in the skin, you can see darker regions of your skin. Okay, let me continue now. Langerhans cells, right? This is going to be in the stratum spinosum. What are these guys? Basically, all they are is they play an important role of immune function, right? They're derived from monocytes, which is a type of white blood cell, and they're derived in the progenitor melanocytes, right? They play a major role in immunity. They recognize antigen to where the skin is exposed. Okay, so they're over here, right? And now with the skin layer, if you get like a little cut or like an exposure or something, right? This is going to say, okay, I'm going to release an antibody, or I'm going to pick up the antibody, and I'm going to send all cells to me to try to help kill it. That's why if like you get a little cut, you see a little swelling, some rashes, right? However, if you have an overexpression of these Langerhans cells, what's going to happen? You're going to see a rash. Why would you have an overexpression? If you have allergies to something, or if like you have, literally allergies. If you have allergies to like pollen or like cats or dogs, etc., your Langerhans cells are overstimulated. So you're overstimulating an immune response, causing a rash. The last cell I want to talk about in the epidermis is going to be the Merkel cells, right? These cells are mechanoreceptors. They're in the basale layer, so they're on the bottom, right, the basal. And all they do, they have a sensory nerve attached to them, right? So the Merkel cell and the sensory nerve, when they're combined, they're called a Merkel cell neurite. It's just a combination of both of them. What do? They feel light touch, right? And they're most common in your fingertips. So if you lightly touch your fingertip, you could feel it. If someone lightly touches like your back or your arm, you might not be able to feel the touch if it's too light. So it's a highly sensitive area. This is called the Merkel cell. So that's the regions of the epidermis. Now let's talk about, let me talk about two diseases of the epidermis. So the first one is melanocyte destructions, right? If you have melanocyte destruction, you're going to get vitiligo. What is this? This is a type of disease, right? Where if you see someone who you have all the same skin color, right? But they have like spotty regions of their body. It's because their melanocytes are not working in those regions. They're not producing what? What are they not producing? They're not producing the tyrosine enzyme. This tyrosine enzyme releases the melanin. They have melanocytes. That's not wrong with their disorder. The disorder is they don't have melanin, melanocytes coming from tyrosine enzymes. That's the spotty skin. Now, another disease is, not a disease, but if you take like whitening creams, what do they try to do? Like if you try to whiten your teeth or something, they're inhibiting this tyrosine enzyme. By inhibiting this tyrosine enzyme, you don't release melanosomes, you don't release melanin. These melanin doesn't go to neighboring keratinocytes, and that doesn't shade the nucleus, which doesn't make your skin change colors. Okay, now, the dermis layer, right? We have two different regions. We have a reticular, and we have a superficial, a papillary. The reticular is deep, it's fibrous, right? This is just a network of elastic fibers. They provide elasticity, so they help our skin stretch, and they have proteoglycans and dermatan sulfate. Okay, now the papillary layer is loose connective tissue. This is vascularized, right? It's got dermal papillae on it, and it has type 1 and type 3 collagen fibers. Type 1 is our main collagen fiber. Also, we have type 7 collagen. This type 7 collagen inserts into the basal lamina to help bind the dermis to the basal lamina. Okay, so remember here, we said we have a hemidesmosome connects the basale layer to the basal lamina. Okay, but for the dermis layer, we have type 7 VII collagen, and he's going to anchor into the basal lamina, and he's going to connect the two cells together. So the basal lamina is like the border, basale is above it, papillary layer is underneath him. Okay, so now, what else is in the dermis layer? We have appendages, right? That's your hair follicles, your hair shaft, right? I said this is not counter, but it aids with stratum corneum. It's like an addition to it. That's what the hair follicle does. Also, we have the skin glands. we have a sebaceous gland, and we have a sweat gland. So for the sebaceous glands, this is a holocrine secretion, right, of sebum. This goes into the hair follicle. What does this mean? So if here's a hair follicle, right, we have a little bulb on him and the hair follicle coming out. If we have a sebaceous gland, he's going to secrete into the follicle. What happens when it secretes into the follicle? It does a couple of functions, right? It protects our hair, it provides antibacterial function to the hair, and it helps smooth the hair. What happens when we have a hormonal disturbance? So like when we're going through puberty, we have hormonal disturbances, which affects the sebaceous glands. So it does like an under or overproduction of sebaceous glands secreting what? Sebum. When that happens, you could see acne, almost like individuals, because they have an incorrect distribution of sebum. Now, sweat glands, we have two types. We have eccrine and apocrine. In eccrine, it's regulated by mainly the palms and the soles of our hands. It helps regulate temperature, right? But it does not need to secrete into a hair follicle. He releases like this watery substance on him, and this will help with evaporation. So if we secrete fluid onto our arms or into our legs, we could help evaporate, or we could regulate our body temperature via evaporation. Now, an apocrine sweat gland, right? It's not really apocrine. This is what the word was. They never changed the name, what it was supposed to be. But it's in the axillary and the perineum, right? So it secretes an odorless fluid, right, near the hair follicle region. And what is this going to do? When it joins with microbacteria, it's broken down and this forms body odor. This is why you sweat, this is why you smell, that's why people are gross. Now, one more function, or one more thing I want to talk about are the nerves. You have two types. You have non-encapsulated and you have encapsulated. Non-encapsulated nerves are basically nerves that just have a free nerve ending, right? So if this was a nerve, right, a non-encapsulated one would be like this. Just like free nerve endings, right? It's not joined together in a good capsule. Okay, so the non-encapsulated ones, you have nociceptors and you have pain receptors, right? This is above the papillary layer. And where is it? It's in the basal lamina, right? Right above at the top. We talked about Merkel cells. So these pain receptors basically feed into Merkel cells. What is another one of these things? The Messener's corpuscle, right? He aids with fine touch. So Merkel cell was light touch. Messener's corpuscle is fine touch. Nociceptor is a heat receptor, right? He sees if it's very hot, like if you touch a stove. This is the guy that helps you move your hand away. This is the first receptor. It would be important for him to be superficial other than deep, because that's like an emergency, drastic thing. If you touch a hot stove, if you touch fire, you need to move your hand fast. Now, you have encapsulated ones, right? This is non-free nerve endings. You have Pacinian corpuscle, you have the Crosser's end bulb, and you have a Ruffinian corpuscle. Okay, so what are they? The Pacinian corpuscle, he's right over here. He's deep. So if he's deep, right, if you push into your body, he detects pressure and vibrations. He's deep. He wouldn't detect light touch if he's superficial, if he's not superficial. The Crosser's end bulb, he's also deep. This is a cold receptor. If you're very cold, this guy's going to feel it first. And also, Ruffinius corpuscle. What does Ruffinius corpuscle do? He is a stretch receptor. So he's deep. If you're stretching your skin at the top region, it's not really going to feel it. But if you stretch the skin on the bottom region, the Ruffinius corpuscle is going to be the one that detects it. So, now, if we have a couple questions I'm going to answer for you in the video. If we have a decrease, right, in dermal collagen, so what was dermal collagen? The layers over here. What is going to happen? Our skin is going to wrinkle because it's not as thick, and we're going to impair our wound healing. So we're not going to be able to wound injuries faster or cuts, etc. Also, what else are we going to do? The thickness of our skin is going to be decreased. What causes this? Things that cause this is old age and extra exposure to UV light. Extra exposure to UV light is going to cause skin aging. There's another thing I wanted to talk about that I didn't, was how the skin divides. So the skin divides, right, fast from the stratum basale, and it starts going upwards, right? It grows superficially. When it grows superficially, at the very top, it starts to lose its nuclei, like we said. So, skin divides and gives more superficially, it eventually loses nuclei. And now we have an epidermal turnover rate, right? So by the time the skin goes from the bottom layer all the way to the top, it takes about 47 days. This is an average. However, it differs in the different areas of our body. If we were to cut like an area of our arm, and then cut an area of our leg, our arm's going to heal much more faster. We heal our upper limbs a lot faster than we heal our lower limbs. Also, if our growing cells, right, exceed the rate of our shedding cells, so if we're growing cells faster than we're losing them, we have a disorder, or we have scaly skin and we're going to lose sensation because we have too much skin. This is called psoriasis. Now, a couple other things. What forms the barrier, the mechanical barrier, for skin to be joined together? This is hemidesmosomes and desmosomes, right? So the mechanical barrier is the hemidesmosomes and the desmosomes. Now, that's only in the epidermis. We also have a mechanical barrier in the dermis. What was that? Collagen. Collagen was the strongest, type 1 collagen was the main one we have, and that forms a mechanical barrier in the dermis. What layer helped with permeability? This was granules, right? Granules in the stratum granulosum, and it was the keratin and the lamellar bodies. So the keratin had the fibular aggregates, and the lamellar bodies helped secrete the lipid to cause a lipid barrier for water. Okay, what cell is responsible for being a barrier to UV light? This would be melanocytes, right? Melanocytes releases the supranuclear cap to shade the nucleus. Okay, which cell provides immunoprotection, right? Which one is an immunological receptor? This would be the cells of the Langerhans cells. Langerhans cells secretes the antibodies, right? It signals an antibody. That's immune response. And which one is, which area here is for homeostasis? This is going to be your sweat glands, specifically your eccrine sweat glands, which release the watery substance to help with evaporation. So that's basically everything for skin histology, and hope it helped.